So who's the spokesperson? Tara, are you doing this or?
Yeah, that was me.
Okay. So what did you discuss, and what's the outcome of your discussion?
So I guess broadly, the short answer is that everyone thought this was a good idea. There was definitely support from the group in general that it would be beneficial, and it was useful for a range of reasons. So we had a list of pros. Most of these were being able to benchmark capabilities. So being able to say to an industry partner or a potential client that you had these capabilities, that you could kind of generate, what's the word? Kind of faith in your capabilities for attracting work. There was a comment about people looking for things like ISO accreditation and just having the network to be able to manage that and demonstrate benchmarking in that kind of environment. More broadly, just kind of being able to share expertise. So picking up on what others can do that perhaps your facility can't, and that could even potentially redirect users to the right place, trying to develop a network of services across the country. So there were definitely lots of pros. I guess the main questions and perhaps what might limit people joining, we're really thinking about how do we define activities that are broad enough that everybody can participate in. So, I mean, I guess, what is the actual benchmarking activity that we're going to define, and how would that suit everybody? I think that was the biggest question that we weren't really sure about. And, if that doesn't suit facilities, then they may not participate. So that's probably the reason why. And so, I get the last part of the conversation then ranged from what kind of activity. It could be as simple as how do you manage samples, to are you actually measuring something? If you're going to measure something, what are you actually defining? And I know this is the discussion that's going on in the other groups, but we weren't quite sure how the actual benchmarking activity would actually benefit everybody, and that's probably the biggest question.
Yeah. Okay. But could this be, okay, no, sorry, go on.
No, sorry, that was pretty much the most of it, I think. I guess in the other con, as everyone would guess, is just the time and the resources that would be required to commit to that, as well as having someone who could manage that effectively and share that to the group. So that was the other major con, I guess.
Yeah. So with regards to the first con about, do you think that multiple samples, multiple benchmarking activities could alleviate that?
Yeah, so we talked about that really briefly, and potentially this could be a multi-year thing where you could opt in and out depending on the type of activity that was being benchmarked. And like I said, we discussed that could range from just data collection, data processing, and not necessarily a physical measurement. We had people who didn't look at protein. So when I made the example, are we measuring a protein ID? That wasn't relevant to them at all. So I think possibly a multi-activity type approach would work.
Look, I think this is a perfect segue, and thanks, Tara. I think this is a perfect segue to the second group, because these guys discussed actually what kind of samples we should analyze. So maybe over to Darren, I believe.
Yeah, thanks, Ralph. And thanks, Tara. Good introduction. Yeah, we had some good discussions. We generally saw value in it as well, although there were some caveats that we talked around a bit. I guess one of the main things we thought was in terms of the purpose, which maybe wasn't our question, but it'd be good if we could use this to show other scientists, biologists, medics, these sorts of people, just how developed things like proteomics and metabolomics are, just to raise awareness and show that you can get robust, reproducible results from different labs. And also to show the broad breadth of things that we can measure with mass spec. And so that is a pro, but also a con in that we didn't see that there'll be one thing you could run across every facility. Everyone, if I skip maybe ahead to the which mode to run, we were thinking people should just run what they run in their lab. So if you run DDA every day, then that's what you run. If you run targeted methods, then that's what you'd run. I don't think people are going to be setting up new methods just for this. But we still thought that would be beneficial to show what's possible on a given sample. The given sample probably just needs to be a simple two-group comparison, because just looking at identifying things in a sample is not usually that useful in biology, but seeing if things are higher or lower tends to be the thing we're mostly looking at. So we didn't come up with a sample, but it's got to be a comparison And we thought, maybe it's a biomedical thing, maybe it's an environmental thing. Maybe there'll be more than one of these, but I think one at a time. In terms of which omics to cover, we're sort of saying, well, multi-omics is the future. So while we do need to do it across all the different omics, with metabolomics, lipidomics, there's more of that issue of everyone running different methods, and you're not going to be able to just put data side by side and say, is it the same? But hopefully you'll see, okay, there's 5 groups have identified these common changes, and perhaps some groups have identified some different changes as well. So that probably leads on to working out what the endpoints are and who's going to analyse the data and how you're going to analyse the data, which is probably another group's thing to discuss. I think that pretty much covers our main points.
Okay, yeah, thanks, Don. I mean, actually, just following up on what you just said, I mean, I guess, and this is what we briefly discussed in the group I was in, is also, it's probably even good if different methods are being used, because if it turns out that one method is being exceptional in identifying and quantifying, proteins and metabolites, this could be potentially also shared in order just to bring other facilities up to speed, or just to use this method instead of their own. But okay, are there any other comments from the audience? What Darren just said?
Sorry, there was one point that kind of follows on that I forgot to raise from ours. One of the other key cons was maybe identifying best instrumentation. So maybe you're not at kind of best practices standards, but it might inform future expenditure, or provide arguments for CapEx investment. And so in that way, that was another a big advantage, I think, for the group too.
Okay.
Well, one point I'd make on that is, like, if you're going to compare samples, I think it'd be most beneficial to use some kind of synthetic sample, or like it could be still biological analytes, but prepared in a way that the changes that people are meant to be picking up are known, rather than just kind of, because you can pick up false positive changes. So we want to be able to say what's a true positive and what's a false positive, because it's easy to find changes if you have systematic errors. So more changes isn't necessarily better. You want to use something where the answer is known in advance.
Actually, a good point. I mean, we were discussing about low versus high complex, and obviously, I mean, most samples we get, in particular, for most omics, it's highly complex and not necessarily chemically defined. But I mean, I think you raise a very good point. If you want to find false negatives or false positives, you have to know what's in there. So it's okay. Okay, so again, perfect segue to the next group. So Sarah, logistics groups.
Yeah, so we've got logistics. So if we were to actually run this, and that was the way we actually looked at this, since we haven't actually done this before, we thought the best way to start would be looking at a simple data-dependent acquisition workflow, but not limiting to that as well. Because obviously different groups have different infrastructure and different workflows that they use as well. With regard to what kind of sample we would do, we'd look at peptides. And the reason we would look at peptides is that that way we are taking out the variability associated with sample prep. Again, we're thinking about the first time we're going to run this workflow. So then that way we're just really going to be assessing the instrumentation and the LC conditions, and ultimately the number of proteins at the end of the experiment. We had a lot of discussion around, how we would actually ship the samples, and we thought that doing a stability test first would be a great idea to sort of get an idea of whether or not the peptides would be okay at room temperature or -80, but obviously there's some cons with that as well. It takes time, and someone has to take the ultimate responsibility of doing that initial work. With regard to the type of sample, I know we just sort of talked a bit before, and we were more looking at a mixture of different types of proteins, so lysates that you commercially buy, such as human form, and you're spiking in at different amounts of human versus E. coli versus yeast. And also you would add in there the synthetic peptides, such as markers or spikeotides or IRT markers, to ensure that you can account for variability that's in there. And the reason for generating such a sample pool like that is that it's not just limited to doing data-dependent workflows. if you wanted to do some quantitative work as well, you can then provide a second sample that have different ratios of each of those different species within the same sort of protein mixtures that you can use multiple years in a row. So depending on what the question that the group wants to actually answer. Which mass spec should be used? Well, that's going to be very variable to which what instruments people do have in their own labs, because there is quite a bit of variability there. Not everyone has Orbitrap. But what we thought would be a good thing to do is to ensure that we do cap the gradient length that you apply, or the LC conditions that are applied for the experiment, to ensure that we're using the same sort of any column of a certain length or at a set length, and then you cap the length of the LC gradient as well. But obviously there's a lot of flexibility with this as well within each of the groups. I mean, this is the first time we would be rolling this out, so we would learn from that as well. So yes.
Awesome, great, thanks, Sarah. Any comments regarding that? I think this pretty much recapitulates what we discussed.
So just there was a good comment from Richard on the chat function about, is there a multi-species equivalent you could do for metabolomics? Which I certainly don't know, but I'm not a metabolomics person.
I guess the question that I have is and it kind of comes from one of ours where we were like looking at whether the vendors should be involved or something like that. In some ways, it'd be nice to have like a standard biological sample that you could either, you know, if you were doing lipids, it'd be nice to know what lipids you could find in it. If you're doing proteomics, you could do proteomics, lipidomics, and metabolomics all on the same sample. That way, you know, you don't have to have a lipidomic sample and you don't have to have a metabolite sample. You want to start with just one thing. Now, I'm not sure what the right sample of that is, but at least that way we would all start with the same thing, and then lipidomics, the people who do lipidomics, they would all run their lipids analysis on it, and they'd be able to compare between themselves, because there might only be, I don't know, maybe there's only 4 labs that are doing lipidomics, maybe there's 20. But the point is they only have one sample that you have to distribute.
It's a good idea. The question is just which, you know, what kind of sample this should be.
Yeah.
It's a good idea. I know what you mean.
The standard one we all use for proteomics is HeLa, right? But, you know, I don't know what the lipid content of that thing is like. Maybe.
No idea.
In metabolomics and lipidomics, I think the NIST plasma 1950, I think it is kind of the standard thing that most people I've talked to in metabolomics, lipidomics have been using. So that's like a standard plasma sample to go with, which I guess you could do proteomics on too if you want to put plasma into your proteomics instrument. But it's quite expensive, I guess, is the only downside of that.
Yeah.
Yeah.
And also the earlier you go in the sample prep, like you send out a bit of tissue or a bit of cell pellet, there's all that extra variability about how good different groups are at extracting it. Whereas at least if you send out the purified or semi-purified biomolecules, they're hopefully more stable, but also it's more likely that at least everyone got the same starting material.
And isn't it important to know that your upstream sample prep is good or bad as well? I mean, if you're, I mean, I guess you're trying to delineate, is the problem on the instrument side or is the problem in the upstream sample handling? I mean, you've got both problems you're trying to solve, right?
Yeah.
So, I mean, I guess if your upstream sample handling's bad, then you're going to come out with a poor answer, and then you're going like, well, geez, we need to figure out where we went wrong. We think the machine's happy, so maybe we can be blundered with our extraction or whatever.
Rounds and rounds of testing.
Yeah, that's a good point. That's a good point.
One of the other pros that we mentioned was the possibility of turning this to something that might end up in a publication, you know, that might benefit people in facilities. And so when you're talking about these kinds of questions that would have broader interest to other groups more widely, these are the kinds of evaluations that if it was to become a larger scale thing could potentially end up in publication or something a bit more broad.
Yeah, the chat is quite active. So Joel just mentioned that we should just provide both and not just peptides or proteins. He's like peptides, proteins, lipids, and milkshake. Also, thanks Matt. So Matt is already offering for making a HeLa E. coli yeast mix and distribute it. Thanks Matt for that.
I don't recall offering, but you know.
Yeah, nice. Yeah, Richard Wilson is also saying maybe we should supply cell pellets and test lysis conditions as well. I mean, this is also something we discussed. But I mean, do you want to? And this is also what Nick just said. I mean, do you want to control or test this as well? Sample preparation, and if something goes wrong, you don't know if it's the instrument or the sample prep. But these are all valid points. Yeah. Okay. Anything to add?
Well, look, I feel like if we go away from a straight peptide mixture or whatever it is, we're not really QCing any one point. You know, if there was a problem with your final results, you don't know if it's a sample prep or the instruments. At least if we go with a peptide mixture, if it's bad, it's your instruments, right?
Yes, I mean the less variability, the better it is. I mean, at least for the first shot. I mean, I agree.
Yeah, I mean, it's two different questions. Are you testing the whole workflow, the service, or are you testing the instruments? And then maybe some facilities here are actual instrument facilities, and that's what you do, and the sample prep's up to the user. So maybe for those people, they don't want to do the sample prep. For others of us, where sample prep is a key part of our service, I'd be really interested to include that in the analysis.
Yeah, Joel is saying agreed, but it could be an opt-in for the raw sample. So you could choose your own adventure, which is certainly possible. Okay. Look, let's, for the sake of time, let's just leave this question and maybe let's more focus towards data analysis. Matt, I think you were in charge of this group.
I certainly was, and it is definitely carrying on from what we have just been discussing, because one of the things that we came up with was the problem mainly of just increasing the dimensions of data that you then need to analyze and account for. So it actually relates to that whole what is the sample question, but it then exacerbates itself when we come into data analysis. So we kind of, there are pros and cons to doing both things when it comes to what we should be providing. I think that we came to the consensus that everyone needs to provide the raw data because we need to have a common analysis pipeline to be able to isolate people's instruments from the bioinformatics. But then equally, it's really interesting to be able to compare people's pipelines to see whether a certain pipeline is actually providing more robust data, or whether people have got a pipeline that actually does things better that other people are not aware of. So it comes back to some things that Tara was saying at the very beginning. One of the outcomes of this would actually be maybe people discover things that they didn't know before about the pipeline that they're using, but they could make it better, or settings that they're using that may be actually compromising the data. But I think the biggest take home from this was that we need to decide how complicated we want this to be, because we could try and be everything. And it would end up with, multiple types of samples being run in different ways on the same machine or different machines through different workflows and then through different pipelines at the end. So that was the main take home of what we were talking about. There's definitely value in letting people do what they want and have it as kind of like an informal, non-competitive competition to see who can get the best stuff out of the sample. So we can all look at each other and go, well, wait a minute, maybe we should be doing that. I guess the least controversial thing was the deadlines for people delivering their data, which was, look, on the receipt of the sample, we should be able to turn this stuff around in 4 weeks to a month. That was pretty universal. Yeah, I think that's about it.
Awesome. Great summary. I completely agree with what you just said entirely. Richard Wilson basically also just said for each pipeline, you need a high enough N, so number of replicates, if you really want to test your pipeline, which is a good point as well. Ben is just saying, agree, we can't do everything though. We should start small and work our way up. Fair enough as well. But look, I mean, if we really want to play it simple, I guess, despite everyone using their own pipelines, which is certainly great, you know, exactly what you said, Matt, we can, you know, find out the best one and then change our workflows. I mean, certainly doing a centralized data analysis might be the easier start, or the simple things to do.
I think that the ABRF studies kind of point the way in doing this, where each year they kind of build on what they did the year before. So we can certainly start out small and say, do a proteomics one for those who are interested and a metabolomics one for those who are interested, and see if we get powerful enough numbers. And then next year go, okay, well, we learnt that. What do we now want to do to be able to extend from that? And then, you know, it basically becomes a rolling thing that we do every year, and every year we generate a new publication out of it to improve the knowledge in the field by the sheer weight of data that we can use for comparison to improve those statistics.
Awesome. Great. Anything to add, or do we want to move to the Darren is just saying, I thought the deadline for data delivery might be one of the interesting outcome variables. True. True. Okay, so look, let's, again, for the sake of time, let's move on to the dissemination group. So Belinda, are you in charge of this? Do you really want to talk about what you guys discussed in this group?
Yeah, thank you. And actually we discussed quite a few things that have been brought up in the other groups as well. One of the main things that we discussed is it sort of depends on what the data is and what the project design is before it gets to the dissemination stage. But we also talked about needing to know what we want to do with the data and with the outcomes of the project, so that upstream we can design it in the right way. So do we want to be able to publish from it? Do we want to get papers from it? And if we do, then that would depend on that would sort of tell us how we need to design the whole project to start with. We also talked about starting out small, and we can sort of grow it from there. We thought the data should obviously be disseminated to the internal participants, anonymised. But we also talked about what Matt just spoke about then, is whether we should have access to the raw data and the full set of data, and thought that definitely we should be able to give the participants at least access to the raw data and the full sets of data that come out of the testing as well. In terms of who should have full access to the data, a little bit along the same lines of what you just said around whether there's that centralized data analysis or whether it comes from each of the groups, and who would have access to the raw data and the full data to start with. We've sort of called this group the organising committee, which would be full of very trustworthy people. There was one suggestion that they might be based in the Cayman Islands. Ralph, you were also nominated as a very trustworthy person. So,
thank you.
So we thought obviously there'd need to be sort of that overseeing group. We thought we could keep it simple and we wouldn't need to involve a third party. Obviously that would be for discussion in the group and who was involved. But if there was that sort of organising committee to keep the data and everything in that central place, and be that contact group as well, then that would probably be okay. But again, we were sort of starting with the start out simple approach and then grow from there. I think the other main thing was collaboration between the groups to determine what the final format would be. So again, just sort of working out what we wanted the end point of it to be, so that we knew what we had to put into the design of it as well. So if we wanted those publications, just being aware of that at the very start, so that we did it in the right way, that we could actually have those publications there.
Awesome, great. Thanks for the summary. Any comment on what Belinda just said? I agree. So can I just ask again about this? Your group discussed that the data needs to be de-identified. What the outcome was? Did I get this right?
Sorry, what was that?
You discussed that the data, or that the results need to be de-identified. So that's one of the outcomes.
Yeah, we actually discussed whether it should be de-identified data or not, and we thought that it should be de-identified, or the lab should be de-identified at least. We did discuss whether you could have an opt-in, so whether you wanted your lab to be able to be identified, you could opt in or out, but we just thought that might become too sort of segmented and and make it a little bit too difficult and fragmented then. But we did think about whether there could be an opt-in to be an identified lab, at least in either in this group in reporting the results, so that then you can reach out to the other groups and you know who to reach out to. But we thought if there's that central contact group or contact person, then yeah,
Yes, ma'am, correct.
that, yeah, de-identify after that. So yeah,
Also,
that's another option.
thanks, Belinda. Super helpful. Thank you very much. Okay, look, last but certainly not least, Nick, you want to give us a brief update on the fishing group, what could be done in the future if successful?
Sure. So we came, we had a discussion about whether the vendors wanted to be or should be in it or out of it, or whether we would involve them and all the rest of it. We kind of, I think we sort of decided that not at the early stage anyway. I mean, we imagined that, suddenly having, one of the application specialists banging on the door going, hi, I have to do a test here or a demo in your lab or something, so that they can then post it. So we thought that maybe not initially that we'd have the vendors involved. Perhaps someone's got other ideas about that. Who should pay for the exercise? We thought about whether, if there is, there are lots of standards on the market from various vendors. We did consider whether or not, Sigma or Thermo or whoever the company is, Avanti or whoever else it is that is making these sorts of standards, would that be an option for us to talk to one of those vendors and see if they'd be willing to help us out there? The advantage of that would be also that it saves money on the shipping, because if it was all being sent, let's say it was Sigma, then the sample could be sent direct from Sigma to each lab, but that way we don't have to try and bring it all into one place, make our own stand, and then separate it all out and send it all off and all that sort of stuff. So the logistics of that would be certainly a lot better. National versus international effort. Initially, international is fine, but initially national just to get it started. But that also kind of leads on to, well, I'll go to the next point, which is just once a year we thought was good because everyone's busy. And actually, I think, certainly for our lab, January is a quieter time. And so that's the sort of time where we'd have time to do it. Whereas if you came to us in October, that would be a disaster for my lab because, everyone's trying to finish up. Where could such an initiative lead? And that goes to the international thing, is whether we join or become the ABRF or something like that. I'm not sure if anyone's ever been to the ABRF. I visited there about 3 or 4 years ago. I went to the ABRF meeting. It was very good. It wasn't a meeting I'd go to every year or anything, but every couple of years it might be worth going to. But I actually got cornered on one afternoon by the president of the ABRF at the time, who was encouraging me to get, Australia a satellite version of the ABRF altogether. So they were very keen to, I think at the time they were telling me they'd set something up in South Africa. But certainly the ABRF is doing everything that we're doing now. They've done this and many more things. If any of you have seen the WINT study at ABRF, that's an amazing effort. And so, there is the possibility that, we could, I don't know what the downside of joining the ABRF or becoming a splinter group thereof or whatever it is might be, but that's another option that we could look at. As for the name, we didn't come up with a better name, no.
Okay. Awesome. Thanks, Nick. Okay, we're pretty much on time. Anything else?
I like it.
People's fond of the APRF, all right. Okay, first of all, thanks so much for all your input. I think it was a super fruitful discussion. I guess if I can summarize what we just discussed, I think the interest is there to kick off such an initiative, and I think that the overall consensus is let's start small and work our way up, and/or maybe go together with APRF. I think it's really good, and I guess all I can say is that both Dan and I we will have afterwards probably another separate meeting and think a little bit more about this. And but if actually quite a few facilities and quite a few of you guys, would do this, I think Ben and I were certainly happy to spend quite some time and get this going. So fantastic. Okay, so with this, if there are no additional comments, then look, let's